Hello everyone, welcome to the channel Microbial World. Today let's learn about bioremediation. Bioremediation is an environmentally friendly, sustainable process that utilizes living organisms, primarily microorganisms and plants, to remove, neutralize or transform contaminants from polluted environment including soil, water and air. So what is bioremediation? It is a process that uses naturally occurring or deliberately introduced microorganisms to break down and consume environmental pollutants in order to clean a polluted site. Now this process it mainly uses microorganisms and also plants, or microbial or plant enzymes are used to detoxify the contaminants in soil and also other environments. And this concept it also includes biodegradation because the contaminants are partially or completely transformed by the microorganisms and plants. And bioremediation it is a waste management technique because it is using naturally occurring biological organisms to convert hazardous substances into less toxic or non-toxic forms. Now let's look into the key principles of bioremediation. Metabolic pathways, catabolism and anabolism. Catabolism is the breakdown of complex organic contaminants into simpler molecules releasing energy. Microorganisms use these contaminants as a source of carbon and energy. Anabolism is the synthesis of cellular components from simpler molecules using the energy produced during catabolism. Microbial enzymes. Microorganisms produce specific enzymes that catalyze the degradation of these contaminants. Bioavailability of contaminants. For bioremediation to occur, these contaminants must be accessible to the microorganisms. So factors such as solubility, distribution and chemical form of contaminants can affect the bioavailability. Environmental conditions. Optimal conditions for microbial activity include appropriate levels of oxygen, temperature, pH and moisture. These factors can influence the efficiency of bioremediation. Microbial communities, diversity and adaptation. A diverse microbial community can increase the likelihood of having organisms capable of degrading variety of contaminants. Indigenous microorganisms are better adapted to local environmental conditions and contaminants. Transformation pathways. Mineralization. Mineralization is the complete degradation of organic contaminants into inorganic substances like carbon dioxide, water and minerals. Co-metabolism. Co-metabolism is a process where a microorganisms degrade a compound while using another compound as the primary energy source. This means that the microorganism does not derive energy or growth benefit from the compound being degraded. Instead, they use a growth substrate as the primary energy source. Reductive dechlorination. Reductive dechlorination is a specific anaerobic process where microorganisms remove chlorine atoms from chlorinated organic compounds, making them less toxic and more biodegradable. Factors affecting bioremediation. The effectiveness of bioremediation depends on various physical, chemical and biological factors. So understanding and optimizing these factors are very important for successful remediation. Environmental factors. Temperature. Most microbial activity occurs within a temperature range of 20 to 40 degrees Celsius. Outside this range, microbial metabolism slows down, reducing the rate of bioremediation. High temperature can increase the volatilization of contaminants, reducing their availability, whereas low temperature can inhibit microbial growth and enzyme function. pH. Bioremediation processes are more effective at neutral pH 6 to 8. Extreme pH levels can inhibit microbial activity and enzyme function. Acidic and alkaline conditions can also affect the solubility and bioavailability of contaminants. Moisture content. Soil moisture levels between 30 to 90% of water holding capacity can favor microbial activity. Too little moisture can limit microbial activity and nutrient transport, whereas excessive moisture can create anaerobic conditions, hindering aerobic degradation processes. Oxygen availability. Oxygen is required for the aerobic degradation of organic contaminants, whereas some contaminants are degraded under anaerobic conditions using alternative electron acceptors like nitrate, sulfate, or carbon dioxide. Contaminant characteristics, chemical structure, complexity. Simple organic molecules are easier for the microorganisms to degrade, whereas complex or highly chlorinated compounds require specialized enzymes or co-metabolism. Recalcitrance. A recalcitrant compound is a chemical substance that is resistant to degradation. These compounds will persist in the environment for longer periods, creating pollution and posing a challenge for remediation. Some contaminants such as polycyclic aromatic hydrocarbons or polychlorinated biphenyls are resistant to microbial degradation due to their chemical structure. Concentration. Very high or low contaminant concentration can inhibit microbial activity. High concentrations are toxic and low concentration may not provide enough substrate to sustain microbial population. Bioavailability, solubility and accessibility. The contaminants must be soluble in water to be bioavailable to the microorganisms. Hydrophobic contaminants are less bioavailable and they require emulsification or surfactants. Contaminants that are trapped in soil particles or adsorbed onto surfaces are also less bioavailable. This can also reduce the rate of biodegradation. Microbial factors. As we have already discussed, diversity and adaptation can influence bioremediation, and also metabolic capability. Microorganisms produce specific enzymes to degrade particular contaminants. These enzymes will facilitate the breakdown of the pollutants to less harmful substances. Nutrient availability. Microorganisms require nitrogen and phosphorus for their growth and metabolism. The addition of these nutrients enhance microbial activity and degradation rate. Trace elements like iron, magnesium and potassium is necessary for microbial growth and enzyme function. An optimal carbon to nitrogen to phosphorus ratio is also required for microbial growth. Imbalances can limit microbial activity and effectiveness of bioremediation. Site characteristics. Soil type and structure. Soil permeability affects the movement of water, air and nutrients, influencing microbial activity and nutrient transport. Soil texture such as sand, silt or clay soil can affect the availability of contaminants and physical environment for microbial growth. Organic matter serves as an additional carbon source for microorganisms, enhancing their activity. Hydrogeology. The movement of groundwater can transport contaminants and microorganisms, affecting the distribution and bioavailability of contaminants. The position of water table can also influence oxygen availability and the type of processes, that is, aerobic or anaerobic process that dominate. Presence of toxic substances. Some contaminants or degradation products can be toxic to the microorganism, inhibiting their activity. Synergistic and antagonistic effect. This means that the presence of multiple contaminants can either have synergistic effect, that is, enhancing effect on the microbial degradation process, or antagonistic effect, that is, inhibiting effect on the microbial degradation. Types of bioremediation. On the basis of removal and transportation of waste for treatment, bioremediation is classified into two different types: in situ bioremediation and ex situ bioremediation. In situ bioremediation. It is the in-site treatment of contaminants using biological agents. In this method, the contaminated material is treated directly at the site without excavation. Under in situ bioremediation comes intrinsic bioremediation, also called natural attenuation. It is a less disruptive and cost-effective method. The method relies on naturally occurring microbial communities. This means that it is a natural remediation process that uses the inherent capacity of microorganisms without taking any engineering steps to enhance the process. And the polluted sites are treated without any human intervention. Monitoring is essential. Now, why monitoring is done? It is done in order to confirm that the natural processes are effectively reducing contamination. In situ bioremediation techniques include bioaugmentation, biostimulation, bioslurping, bioventing, and phytoremediation. Bioaugmentation involves introducing specific strains of microorganisms that have the ability to degrade soil and groundwater contaminants. In this technique, strains of natural or genetically engineered bacteria with unique metabolic profiles are added to the contaminated site in order to supplement indigenous microflora and speed up the biodegradation process. Bioaugmentation is mainly applied for chlorinated contaminants and petroleum hydrocarbons. Microbes cannot use pollutants as the only source of energy. They also have to be supplied with nutrients. So, biostimulation specifically focuses on adding nutrients, for example, nitrogen or phosphorus, to enhance the activity of indigenous microorganisms. It is a very simple and cost-effective method to boost microbial degradation. The method requires monitoring to avoid over-fertilization. It is a common remediation approach against petroleum pollutants in soil. Bioslurping. It is also called multi-phase extraction. It is a technique used to clean up contaminated groundwater and soil, particularly where hydrocarbons like petroleum are present. It combines the elements of two methods, bioventing and vacuum-enhanced extraction. A vacuum pump is used to draw out contaminants from the soil and groundwater. Oxygen is introduced to stimulate naturally occurring microorganisms that break down the contaminants. So the vacuum pump slopes the liquid contaminants and vapors, while the introduced oxygen enhances the biodegradation process, making bioslurping a very effective method. Bioventing is the most common in situ treatment. It involves injecting air or oxygen through wells into the contaminated soil to stimulate the aerobic degradation of pollutants by indigenous microorganisms. It is very effective for the remediation of petroleum hydrocarbons and other volatile organic compounds in soil. Phytoremediation is the process that uses various types of plants to remove, transfer, stabilize, or detoxify contaminants in soil and groundwater. It is effective for heavy metals and organic contaminants like pesticides. There are several different types of phytoremediation mechanisms such as phytodegradation, phytostabilization, phytoextraction, rhizofiltration, phytovolatilization, and phytostimulation. In phytodegradation, the plants actually metabolize and destroy contaminants within the plant tissues. Phytostabilization is the process where chemical compounds produced by the plants immobilize contaminants rather than degrading them. Phytoextraction is also called phytoaccumulation. It is the process where plant roots absorb contaminants along with water and nutrients. The contaminant mass is not destroyed, but it ends up in the plant shoots and leaves. Rhizofiltration is similar to phytoextraction, but the plants used for cleanup are raised in greenhouses with their roots in water. Phytovolatilization is the process where plants take up water-containing organic contaminants and release the contaminants into the air through their leaves. Phytostimulation refers to the enhancement of plant growth and development through the use of biostimulants. These biostimulants can be natural or synthetic compounds such as plant hormones or beneficial microorganisms such as mycorrhizae or rhizobacteria or other organic compounds. Ex situ bioremediation. In ex situ bioremediation, the contaminated material is removed from the site for treatment. This method allows better control over environmental conditions, but it can be more expensive and disruptive. Ex situ bioremediation techniques include biofiltration, biopile, bioreactor, composting, and land farming. Biofiltration is a technique that uses living organisms, primarily microorganisms, to remove contaminants from air, water, and wastewater. Biofilters consist of a bed of organic material. This bed media provides a surface for microbial growth and a medium for contaminant degradation. Common materials include compost, soil, peat, wood chips, and various other synthetic materials. The contaminated air or water passes through the bed media, allowing the microorganisms within the bed to degrade the pollutants. In biopile method, the contaminated soil is excavated and piled above ground. The piles are aerated and irrigated to enhance microbial degradation. Nutrients and oxygen is also often added to stimulate microbial activity. Bioreactor is an engineered system involving series of biological reactions. The pollutants are fed into a bioreactor vessel for their degradation to facilitate the growth of biological mass. In bioreactors, the contaminated material is placed in a controlled environment where conditions like temperature, pH, and nutrient levels are optimized for microbial degradation. Types of bioreactors include slurry phase bioreactors for liquid or slurry-like materials and solid phase bioreactors for solid materials. Composting is a biological process that converts organic waste materials into a stable, nutrient-rich soil amendment known as compost. The organic waste materials can be green materials such as grass clippings, vegetable scraps, coffee grounds, and manure, or brown materials such as dried leaves, straw, wood chips, or paper. The process involves aerobic decomposition of organic matter by microorganisms such as bacteria and fungi. It is an environmentally friendly way to recycle organic waste and improve soil health. In land farming technique, the contaminated soil or sludge is evenly spread over the treatment area in thin layers. The treatment area is an area of land that is specifically designated and prepared for land farming. The soil is periodically tilled to aerate and mix the contaminants to enhance microbial activity and degradation. Liners and drainage systems are installed to contain contaminants and prevent leachate migration. Microorganisms involved in bioremediation. Bacteria. Pseudomonas species are known for their ability to degrade petroleum hydrocarbons and polycyclic aromatic hydrocarbons. Bacillus species produce enzymes that can break down complex organic compounds including pesticides and herbicides. Alkalogen species are used in the treatment of industrial wastewaters and contaminated soil. They can degrade aromatic compounds and other organic pollutants. Mycobacterium species degrade polycyclic aromatic hydrocarbons and other complex hydrocarbons. Rhodococcus species degrade a wide range of hydrocarbons, including chlorinated compounds. Nitrosomonas and Nitrobacter species play a key role in the bioremediation of nitrogenous waste. They are involved in nitrogen cycle converting ammonia to nitrate. Dihalococoid species are effective in the bioremediation of trichloroethylene and other chlorinated organic solvents. Acinetobacter species degrade hydrocarbons and other organic compounds. Fungi. Phanerochaete chrysosporium, white rot fungus. It degrades lignin and a variety of pollutants including polycyclic aromatic hydrocarbons and pesticides. So they produce extracellular enzymes like lignin peroxidase and manganese peroxidase that break down complex organic molecules. Trametis versicolor has the ability to break down dyes, pharmaceuticals, and other recalcitrant compounds. Aspergillus niger degrades heavy metals and various organic pollutants. Penicillium species are also effective in the bioremediation of soil contaminated with heavy metals and organic pollutants. Archaea, methanogens, example Methanosarcina species. They play a key role in the degradation of organic pollutants in anaerobic environment, contributing to the breakdown of complex organic matter and production of methane. Halophilic archaea, example Halobacterium species. They degrade organic pollutants in high salt environment. So these organisms are adapted to extreme conditions capable of carrying out bioremediation in saline and hypersaline environment. Advantages of bioremediation. It is environmentally friendly. Bioremediation uses natural processes to degrade pollutants, thus minimizing environmental disruption and also avoiding harmful chemicals. Cost effective. Bioremediation, it is cheaper than traditional methods like excavation, incineration, and also has a reduced waste disposal cost. Bioremediation is versatile, meaning it is effective against different contaminants like hydrocarbons, heavy metals, and also adaptable to different site conditions. It is sustainable, meaning it requires less energy and uses renewable biological processes. It enhances soil health. Bioremediation improves the soil structure and fertility, increasing microbial diversity and activity. Safe and non-toxic. So bioremediation, it produces minimal harmful byproducts, thus reducing the risk of secondary pollution. And it has complete degradation potential because bioremediation can fully break down the contaminants into harmless substances, thus providing a long-term solution. And it is applicable to diverse environments like soil, water, and air. It is also enhanced by technological advances, which means it can be improved by methods like biostimulation, bioaugmentation, genetic engineering, thus increasing the efficiency and effectiveness of bioremediation. Disadvantages of bioremediation. It is limited to biodegradable contaminants, which means it is ineffective for non-biodegradable pollutants or very persistent contaminants. And it is a slow process. It can take months or years for complete remediation depending on the contaminant and site conditions. Environmental sensitivity. Bioremediation can be affected by factors like temperature, pH, moisture, or nutrient availability. Incomplete degradation. Sometimes only partial degradation occurs, and that will result in intermediate products that can be harmful. Site-specific conditions. Bioremediation requires specific conditions for optimal microbial activity which may not be present in all contaminated sites. Monitoring and maintenance. It needs regular monitoring and also nutrient or oxygen addition in order to ensure continued microbial activity. Bioremediation processes also has the potential to produce toxic byproducts that might need further treatment. Variable effectiveness, meaning it cannot be equally effective against all type of contaminants or in all type of environmental settings. Applications of bioremediation include soil remediation. It can clean contaminated soil, especially soils affected by oil spills and pesticides. Water remediation. Bioremediation can treat polluted groundwater and surface water from industrial and agricultural activities, and it can also clean marine oil spills. Wastewater treatment. It can reduce organic waste and contaminants from municipal and industrial wastewater. Air pollution control. Bioremediation can remove harmful gases and volatile organic compounds from industrial emissions using biofiltration technique. Landfill leachate treatment, which means it can treat the harmful substances in the liquid that leaches from landfills. In mining sites, bioremediation manages and reduces acid mine drainage. Whereas in military sites, it can degrade explosives and residues in soil and groundwater. Now, in case of agricultural runoff, bioremediation can remove excess nutrients and pesticides before they contaminate water bodies. Brownfield redevelopment, which means it cleans up the contaminated site, making them safe for new development and use. So this is all about bioremediation. Thank you for watching. Do subscribe the channel for more informative videos.